5 prime, 3 prime. And then you will now have your replication. So as your helicase moves along the DNA structure, it will unzip your DNA. It will unzip your DNA to double helix. So first is your helicase. Next is to keep your DNA this way. We use your single-stranded binding proteins. Single na stranded pa. Yung pangit. So binding proteins. Joke lang. We all belong to the same. Okay, so single-stranded binding proteins such that our DNA double helix will remain this way. So helicase, binuksan, single-stranded binding proteins. And lastly, papasok yung enzymes natin for priming, which is your primases. So your primases create short RNA molecules along the DNA strand. So, kunwari, ito yung primase natin. It's going to create short RNA primers. So, pupunta siya sa isang exposed DNA strand natin. It creates RNA primers. So, remember, if this one runs in the 3' to 5' direction, ano yung direction ng primers natin? Anti-parallel din.
Anti-parallel.
5' to 3'. So, eto, lalagyan din natin ito ng primase.
3'.
So, ano yung direction niya?
3 to 5'.
3' to 5'. Correct? So, we are now ready for the process of elongation. So, ano ulit yung ginawa natin? In-unzip, ni-remain na unzip, and then pinrime. Okay. So, in the process of elongation, this is where our major proteins, or rather, our major enzyme comes in, which happens to be DNA
Polymerase.
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 Okay, your DNA polymerase will try to scan your DNA, and then, remember saan nga ulit tayo dumadagdag ng nucleotides? In the 3' OH group. So, saan mangyayari yung synthesis? Let's say, this is your DNA polymerase. So, it will move along and create. Tama? Yes. So, ang problema nito guys is that in this specific strand, saan yung 3' end natin? So, saan mangyayari yung elongation? It's towards this direction. So, anong mangyayari dito? In short, this is your lagging strand and this is your leading strand. Familiar with the terms?
Yes.
Leading strand. So, the whole point bakit may leading and may lagging strand is because of the direction of your polymerization. So, kunwari, eto si DNA helicase natin, patuloy siyang nag-unzip. So, nag-unzip pa rin siya. So, ito, unzip DNA ito siya. So, in this specific strand, the direction of synthesis will be continuous. Tama? Because it runs in the 5', 3' direction. Whereas this one, so ano mangyayari dito? Magkakaroon tayo ng fragment. So what needs to be done is that kailangan ulit pumasok si primase para i-prime to siya ulit.
Okay.
Remember? And
Para
then,
maganda.
so this region over here and this specific region creates this gap. This is what you know as your Okazaki fragment.
Okazaki.
Okay.
Okazaki fragments, Suzuki fragments, mayaman yung kapon sa mga sasakyan. So ito, so OH group runs in this direction, so your, nandito yung 5', 3', so dito papunta yung synthesis natin. So in short, in your lagging strand, synthesis occurs na pahinto dito. Whereas in your leading strand, it occurs very continuously. So far, so good?
Yes.
So this is the whole point why DNA replication is said to be semi-discontinuous. Kasi medyo pahinto-hinto siya. Bakit medyo? Kasi ito pahinto-hinto, ito diretso. Gets? Okay, so this explains why you have your leading strand and your lagging strand in the process of replication. Question so far? Kakapi lang ako. Okay. So, replication is the process by which DNA creates identical copies of itself and occurs when? During S phase. Okay, so yung S phase, ito siya. Of your interface. Okay? So, characteristics of replication, na-discuss na natin siya. Semi-discontinuous, ito yung sa kanina. Okay, leading strand, lagging strand, yan siya kanina. Ito, to visualize everything. So, this is your leading strand. So, this becomes your lagging strand.
Lagging
Because
strand.
if this is your replication fork, in your lagging strand, saan papunta yung replication? Opposite of the synthesis. Whereas in your leading strand, it's toward your replication fork. Ayun. So, semi-discontinuous siya. Nice, no? Okay. Saya magtulong kapag advanced for class kasi hindi na kailangan isa-isahin. So happy. Okay. So, proceed na tayo sa transcription, guys. So, yan na yung replication. By the way, how does replication terminate or how does it end? Once everything has been fully replicated. Because remember, you can only replicate your DNA once during the cell cycle. Okay? Okay yan? Okay. So, this is some RNA primers pa. Alam na natin yan. The best students ever. Woo! Okay. So, punta na tayo sa transcription. So, kung sa replication, in-ensure natin na yung genetic material natin is double. In the process of transcription,
Ito yung pinapanood ko sa YouTube na
in the process of transcription, what happens here is that we are now going to express our genes. So, sa transcription, let's try to recall again our genetic structure. So, if this is your DNA, tapos let's say for example, you will have certain regions there which code for a specific trait. Let's say this one codes for brown eyes. Let's say this one codes for your anti-cancer gene. Let's say this one codes for let's say anti-UV gene. Yan siya. So yung nangyayari dito is that not all genes are being expressed at the same time. So let's say for example, I have this stretch of genetic material in front of me. So, kunwari, naglakad ako sa araw, tapos na-expose ako sa sobrang lakas ng init ng UPLB. So, ano yung kailangan ko dito? Gene for brown eyes? Gene for anti-cancer? Or gene for anti-UV?
Anti-UV.
Anti-UV, very good. So, in short, gene expression only occurs when the cell needs the gene products. Do you follow? Whereas, your replication
Purposive,
will
sir.
occur every single S phase of your cell. Okay? So far, so good? So,
Good so far.
paano ito nangyayari?
So good, so
So, this is done by the process of transcription. Because, you need to create your protein which has necessary anti-UV characteristics kasi nga nagpa-araw ka, di ba? So yun, ano na nangyayari dito? So the process of transcription still requires initiation, elongation, and termination. Sila na naman. Sila na naman. Okay, so ano nangyayari dito? During the process of initiation, we will be using the sigma factor. What happens here is that your sigma factor will try to take a look at the gene which it needs to express. So, it appears that for that specific moment in time, we will be needing the gene for anti-UV. So, what happens here is search to ng sigma factor. Okay, andito yung gene for anti-UV. What it does is that this one looks for the needed gene. And then, it will recruit the major enzyme. So, ano nangyayari dito? So, I need this gene. So, nahanap ko na siya. Dito ko papapasukin yung major enzyme natin. The major enzyme happens to be RNA polymerase. So, kung DNA polymerase kanina yung required sa process of replication, this time we will be needing your RNA polymerase. So far, so good? Do we follow? So, yung gagawin ng sigma factor is, okay, I need this gene, RNA polymerase, dito tayo. Kasi itatranscribe natin to, gagawa natin to ng photocopy. So, yun. After RNA polymerase has been docked in the region before the gene, or known as your promoter sequence, your RNA polymerase will then proceed to transcribe the gene. It will now transcribe the gene. So let's say for example, yung gene natin is composed of, let's say, ito, A, B, C, A, G, G, D, G, C, E, kunwari, ito yung ating gene which codes for the anti-UV protein. So, T, A, C, C, A, C, G, A, D, D, ayun. So, yung RNA polymerase natin, andito na siya sa region before ng gene. And what it does is that it tries to take a look at one specific region, or rather at one specific sequence lang. And this is our
Anti-UV.
sense strand, and this is our anti-sense strand. Later we will understand bakit may sense saka anti-sense. But for the process of transcription, the RNA polymerase will use the anti-sense strand as template. So, yung RNA polymerase mo, mag-proceed yan siya. So, mag-create siya ng RNA. So, therefore, T pairs with A pairs with U. Very good. This time, uracil na because remember, it now synthesizes RNA. C pairs with G. A pairs with U. Very good. Ayun. G, C, U,
Bakit yun na?
U,
Kasi nga RNA na yung ginamit.
A,
Yung RNA polymerase.
Ano
So
yung
in short, we
uracil?
are now creating
Kasi nga magkakama.
your RNA molecule
Yun, di ba?
out of your DNA structure. So what happens is that once your RNA polymerase is done synthesizing your RNA molecule, so elongation na yun, bale, papasok na tayo sa termination. So for termination, kung may sigma factor, meron tayong rho factor. What your rho factor does is that i-attack niya yung ating RNA polymerase. So kapag na-attack niya yung RNA polymerase, matatanggal siya sa ating gene such that etong sequence na to, matatanggal din siya and your DNA is restored. Do we follow?
Yes.
Okay, so ulitin natin. In the process of initiation, sigma factor recruits your RNA polymerase. Tapos saan niya idadock? Sa
Before.
promoter region. Very good. And the RNA polymerase will proceed with transcription such that it uses which specific template? The antisense strand to create your
Sequence.
RNA. And once your RNA polymerase has reached the end of the gene, anong mangyayari? Sino papasok? Rho factor destabilizes the whole transcription, the whole transcription mechanism such that it liberates your RNA. Ito, mawawala siya. Your DNA will be restored.
Ang balik ang video.
Okay?
Wow.
So far, so good?
Hindi alam yun.
So, ganito yung nangyayari sa, kunwari, kung yung tao
Ano yung sorry yan?
nagkakaroon ng problema sa kanyang cells and the cells are starting to become tumors. So, it will express anti-cancer genes.
Pinabasa ko
And
ng-
consecutively, since your eyes are always color brown, so it will always express the gene for
ano
eyes.
kasi to?
So the cell will express the gene whenever it feels the need to express the gene.
Akala ko sa transcription. Puro RNA polymerase na yung laging enzyme na ano.
Sir, may so much. 5 minutes. See,